Okay, so this lecture is going to pick up where we left off on Thursday, where we finished talking about how the DNA is modified and interacts with each other, with the other sister chromatids to affect homologous recombination. Now we're going to talk about the proteins that facilitate that. So we talked about how the DNA moves. Now let's talk about how proteins process the DNA to make that happen. So in bacteria, the bacterial recombination machinery that's critical are the Rec-BCD proteins shown here, as well as the Rec-A protein, which we're going to get to in a minute. Rec-B, C, and D form a heterotrimer. Rec-C, shown here in kind of this pink color, is in the middle, and it contains a pin domain, that's what it's called, that's a protuberance that physically breaks the hydrogen bonds in the Watson-Crick base pairs. Rec-D has 5' to 3' helicase activity, while Rec-B has 3' to 5' helicase activity. So that right there will tell you that the Rec-B helicase domain here is going to be functioning 3' to 5' to pull this strand of DNA through, while the Rec-D is going to be working on the complementary strand, pulling 5' to 3'. Now Rec-B has an additional domain called the nuclease domain, and when we cut away from, like, do a kind of a cutaway view of this complex with DNA bound, what I hope you can appreciate is right here is that pin domain of Rec-C that is splitting the double-stranded DNA into single-stranded. Rec-B, 3' to 5', is acting on this single-stranded DNA to pull it through. Rec-D is working 5' to 3' to pull this strand through. And both of these single-stranded DNAs they exit this complex near the nuclease domain of Rec-B. Now, I alluded earlier that there's 3' and 5' end processing to generate the 3' single-stranded overhang that's going to execute the strand invasion, which is what we were working with in the holiday junctions last week. This is the complex that makes that. And the way that it makes it is like this. So we have our Rec-BCD complex here that has bound to the double-stranded break and is moving its way, exerting its helicase activity and splitting the DNA along the double-stranded DNA. Now, it's a helicase, so it executes this helicase activity in an ATP-dependent manner. And what happens is the helicase of Rec-D moves slightly faster than the helicase of Rec-B. The nuclease domain of Rec-B is alternating between cutting the 5' end and cutting the 3' end of the complementary strand. Now, our diagram is really helpful in that it's modeling the rate at which these DNAs are cut. There are less frequent cuts along the 5' end, and that is because this DNA is being pulled faster than the 3' strand. So imagine you're going down a highway and you're throwing something out of the car at a regular pace, your arm's moving at the same pace. If your car is going fast, then you're going to skip a bigger chunk of land outside the car as you're throwing things out than if you were going slower. The analogy that I really like for this is a float in an Independence Day parade. Those floats go really slow so they can hit all of the kids along the parade route. If the cars speed up, then they're going to be regions of kids that don't get any candy because the car is moving faster. Okay, so this nuclease domain is acting on these two strands. When the complex reaches a chi site, which stands for crossover hotspot instigator, what happens is the nuclease domain, the helicase domain of Rec-B is going to bind to this chi site and basically sit down and no longer exert the helicase activity, allowing the DNA to leave the helicase domain. However, it's still going to exert helicase activity, it's just not going to let it leave. And so that's going to generate a loop of DNA, not unlike what we saw at the replosome. However, the REC-D helicase activity is not going to be affected by the Chi site, and so it's going to continue to pull the DNA, it's going to continue to get chopped. And so here what we're going to end up with is we're going to end up with a short 5' end and a long 3' end. And this long 3' end is going to be our invading strand into the sister chromatid to create the displacement loop. To create the displacement loop, however, we need the function of another protein called REC-A. Now, what REC-A looks like is this. It has a core domain with an N-terminal domain and a C-terminal domain. And these two domains here, the N and the C, are really important for interaction with other REC-A molecules. So as you can see up here, the REC-A molecule oligomerizes onto the single-stranded DNA, shown here in blue. And with different intensities of green, you can see different molecules of REC-A. The N-terminal domain of one REC-A molecule interacts with the C-terminal domain of another molecule, and in this way it wraps around the single-stranded DNA, forming a helical filament. Now this serves two purposes. The first is that it protects the single-stranded DNA, but more importantly, and we can see this in an electron micrograph, is that it takes our naked, kind of disordered, unstructured DNA and really forces it into a very straight, rigid complex. And this rigidity is going to be really important for facilitating the strand exchange. So the REC-A filament is going to be assembled on the 3' single-stranded DNA in one direction. So it's going to bind on the 5' side of our 3' single-stranded overhang. And once one molecule binds, then we're going to get rapid extension or oligomerization of additional REC-A molecules in the 5' to 3' direction. So we're going to be adding new molecules on to the 3' end of the DNA. Similarly, disassembly is also going to happen in the 5' to 3' direction, so that the last molecule to come off is going to be the one that's at the 3' end. And this nucleation is slow for a reason. When we have single-stranded DNA in the cell, we want to protect it. And so during the synthesis of that 3' single-stranded overhang by the REC-BCD complex, the single-stranded DNA is going to be bound in bacteria by the single-stranded binding protein. This is pretty difficult for REC-A to displace initially. And so a complex formed by REC-F, REC-R, and REC-O, sometimes referred to as the REC-FOR complex, is going to bind at the single-stranded double-stranded junction. And that's going to recruit the protein REC-A, which will then bind near the REC-FOR complex at the single-stranded double-stranded junction. And then the rapid polymerization of REC-A filament on the single-stranded DNA will be enough to displace the single-stranded binding protein. It was a really nice paper about 5 years ago, well, 7 years ago now, that that articulates and describes this this tiered nucleation process. Okay, so now that we have our single-stranded DNA, the 3' overhang, bound by our helical REC-A filament, let's talk about how the strand exchange or strand invasion is facilitated. So we're looking down the axis of the DNA and the REC-A filament. So our REC-A is on the outside of the DNA. And here in our primary binding site is the single-stranded 3' extension. This complex is going to bind to double-stranded DNA. In our perpetual example, the intact double-stranded DNA of the sister chromatid that did not suffer the double-stranded break. So when this double-stranded DNA binds in the secondary binding site, there's going to be a little bit of torsion induced on the double-stranded DNA, the way that it's bound by the REC-A filament, this helical filament, that's going to favor the breaking of the hydrogen bonds between the sister chromatid DNA and favor the formation of base pairing between one strand of the single-stranded or the double-stranded DNA and our single-stranded 3' overhang invading strand. So basically what's going to happen is this guy binds, but it's in an energetically unfavored conformation, and the lower energy conformation of this complex is going to be where the single strand that invades base pairs with one of the two double strands. Okay. So that's our strand invasion, and then the DNA polymerase comes on, executes the replication to fix the double-stranded break. Now let's assume that we are in double-stranded DNA break repair, and we have a holiday junction shown here. Here's all blue, here's all red, here's a red-blue heteroduplex, and here's a blue-red heteroduplex. We need to resolve this holiday junction. And if you take your pipe cleaner model and open it, you'll see that this is the same conformation of double-stranded DNA as what you can make with your pipe cleaners. So the resolvase complex is composed of 3 proteins: RUV-A, which is a tetramer that binds to the holiday junction core; RUV-B, which is a helicase that facilitates branch migration. And what that's going to do is it's going to move the DNA sequence through the holiday junction. And this serves to provide some genetic diversity, so that if you have a hot spot, because that chi site is always going to be at the same place, you don't want to always exchange at the exact same place, because then you're limiting your genetic diversity. And so RUV-B is moving the DNA through this holiday junction to basically randomize, you could think of it that way, where the holiday junction resolution is going to be. And then what happens after some branch migration is going to be the protein RUV-C is going to come on 2 molecules, and it's going to bind the holiday junction. RUV-C is the resolvase. It's going to execute an endonucleolytic cut at opposite sides of the DNA and religate it together. And so this right here is the mechanism by which we either have an X cuts or we have cuts at the Y sites. So this is again where those pipe cleaner junction models can come in handy, where we're cutting across and ligating this side here to this side here, and ligating this side of the cut to this side of the other cut to make molecules that look like this, where we're going to end up making molecules that will be ligated. One will be an intact blue, and one will be partially blue, partially orange. One will be an intact red or orange, and the other will be partially blue, partially orange. So if the RUV-C molecules bound in a 90-degree angle, so instead of binding here, they bound in this direction, we would get the opposite cut to resolve the holiday junction. Okay, so I want to talk practically right now. A lot of you have done some molecular biology in either teaching lab or your research labs. And what I'm showing you here are several E. coli strains that are used in molecular biology and biochemistry labs. These are their genotypes, and they can get pretty hairy and very detailed. But what I want to note for the 4 that I have highlighted in bold, they all have deficiencies in their REC-A protein. These 2, the Rosetta and the BL21, do not. And so what I want you to think about, and what you know, and you guys can talk about this in class during the problem set, is why would it be beneficial to have a recombination deficient strain of E. coli? And in what situations do you not want a recombination deficient E. coli? So we'll come back and review this on Thursday, but I just want you to think about what things do you do with bacteria that would be helpful to have defective recombination, and what kind of processes or applications do you use bacteria for where you would want recombination to be active. Okay, so let's talk about one example of this. In mitosis, double-stranded breaks are damaged all the time. However, in meiosis, there are many examples where double-stranded breaks are intentionally formed by endonucleases that are expressed specifically during G2 or S and G2 to increase your genetic diversity. And this is getting into a little bit of genetic variation. So in mitotic recombination, here I'm showing you the eukaryotic equivalents of REC-A and REC-FOR, RAD51, 52, and BRCA2. Those of you who work in Dr. He Lewis's lab will recognize these genes for sure. And we can repair our double-stranded break through two pathways: synthesis-dependent strand annealing, where we copy a little bit of sequence from the intact sister chromatid and then re-anneal the broken DNA strands and then synthesize through, which will not result in crossover. Or through double-stranded break repair, where we have an invading strand that does not get remodeled, and we end up with double holiday junctions. So this was our site of damage, and we have a holiday junction on the left, and we have a holiday junction on the right. And one of the things you'll be doing in your problem set in class will be to understand how the combination of cuts, whether REV-C cuts at the X or the Y here, and whether REV-C cuts at the X or the Y here, how that results in either genetic crossover or non-crossover. So you'll work through that in your problem set in class. However, the whole term of genetic crossover comes from things that we saw in karyotype spreads or metaphase spreads, where we actually see what looks like flopping over of one sister chromatid and the other, and exchange of chromosomal material. And so what happens, this is beneficial, right? You guys learned this in genetics, that instead of having the big A and big B alleles linked together, we can mix things up a little bit and have the big A linked to the small b allele. And when that happens during the first phase of meiosis, then we can end up with a big A and a little b allele in the gamete, thereby giving us some considerable genetic variation in our population. In yeast, the way that this is facilitated is through the endonuclease called SPO11 that's expressed during meiosis. It has a tyrosine in its active site. And this hydroxyl here acts as a nucleophile to execute a double-strand or a nucleophilic attack, resulting in a double-stranded break in a targeted spot of the genome. And this transient covalent intermediate then becomes a substrate for what we call the MRX complex, comprised of the proteins MRE11, RAD50, and XRS2, which come in and function as helicases and endonucleases to remove the SPO11 conjugated sequence. And then this small 3' overhang with the 3' hydroxyl is further processed by exonucleases called SAE2, and then helicases and exonucleases called SGS1, DNA2, and or exonuclease 1. And these guys, including DMC1 and RAD51, which are analogous to REC-A, now those come in and bind to our 3' single-stranded overhang, which can form, where it can invade and form a displacement loop, and double-stranded break repair can go on, as we discussed earlier. Now, double-stranded break repair and synthesis-dependent strand annealing are both dependent on the presence of a functional polymerase. When homologous recombination is not functional, either because REC-A isn't present or polymerases aren't being expressed, the cell does have a backup way to address double-stranded breaks, and that's through a process called non-homologous end joining, or NHEJ. And what happens in that case is a heterodimer called Ku70 and Ku80 binds to either side of the double-stranded break. And you can see here that the double-stranded DNA is kind of threaded through a channel here between the Ku70 and the Ku80 protein in this dimer. Now, Ku70 and Ku80 are going to recruit some more proteins, including DNA PKCS and Artemis. These proteins function to degrade the DNA, the double-stranded DNA, and basically they're just chopping it back until this complex is able to find about 2 to 5 nucleotides that are complementary to each other. During this widening and this search for a very short region of complementary DNA, we are losing chromosomal sequence, which is very risky But much like the translesion polymerases, this is the only chance that the cell has to repair this double-stranded break and have a chance at replicating and continuing through mitosis. It can't do this if it has a double-stranded break. So even though this process is coming at a potentially significant and damaging cost because we're losing chromosomal sequence, it's a lot better than not replicating at all. So once the PKCS and Artemis complex identify a short region of complementary sequence, then additional proteins, XRCC4, XLF, and a DNA ligase will come in, fill this in, and ligate the DNA together. And so we will have repaired the DNA in the sense that it will no longer have a double-stranded break, but we will have lost a considerable amount of sequence around the site of the original double-stranded break. And so of all of our DNA repair mechanisms, NHEJ is the most highly mutagenic, followed shortly or right behind it is the translesion polymerases. Okay, so on Thursday, we're going to come back and talk about site-specific recombination that uses the synthesis-dependent strand annealing and double-stranded break repair mechanisms to affect several controlled processes in cells, like mating type switching in yeast, switching between MAT-A and MAT-alpha. We're going to talk about DSBR-like site-specific recombinations, including Cre-LOX and FLIP-FRT systems, which were used to make the first transgenic animals in the late 70s and early 80s. And then more modern DNA modification and gene therapy approaches that incorporate a site-specific endonuclease. The TALENs are transcriptional activator-like effector nucleases. ZFNs are zinc finger nucleases, and then you all have heard of CRISPR-Cas9, which is now the gold standard for doing site-directed or sequence-directed cleavage of the DNA. On Thursday, I'll touch lightly on transposons and other mobile elements in DNA, and then finally we'll talk about how these kinds of site-specific recombination events are used by B cells to generate immunoglobulin diversity.